In 1952, relatively little was known about genetic transfer, DNA, or gene replication. Most scientists at the time believed proteins and not DNA were responsible for genetic inheritance. Martha Chase and Alfred Hershey conducted a series of experiments that helped disprove this notion and establish DNA as genetic information. To do this, Chase and Hershey used bacteriophages, a type of virus that are capable of infecting bacteria. These viruses contain a protein coat called the capsid and a nucleic acid core which contains DNA or RNA. First, the phage attaches to the surface of the cell. Then, viral DNA enters the host cell. Phage DNA now replicates and more phage proteins are made. Then, new phages are assembled. Finally, the bacterial cell undergoes lysis, releasing newly made phages into the environment. Crucial to this experiment is the fact that phosphate is highly concentrated in DNA, but not proteins, while sulfur is highly concentrated in amino acids, but not DNA. This enabled Hershey and Chase to track the presence of DNA in proteins by using radioactive isotopes of these elements. Hershey and Chase prepared 2 batches of phages. In each batch, the phages were produced in the presence of a specific radioactive element. The first sample was produced in the presence of 35S, a radioactive isotope of sulfur. The second sample was produced in the presence of 32P, a radioactive isotope of phosphorus. After the phages had incorporated these isotopes, they were allowed to infect samples of E. coli, and the resulting cultures were blended and then put through a centrifuge in order to cause the phages and bacterial cells to separate. Movement through a centrifuge causes heavy particles to move down and lighter particles to move up, which is how separation occurs. In this case, the detached protein shells of the phages rose to the top, while the bacteria sank to the bottom of the test tubes. Hershey and Chase found that 32P, indicative of DNA, appeared in only the bacterial pellet, while 35S, indicative of protein, was only found in the supernatant. Bacteria infected by 32P phages remained labeled, whereas bacteria infected by 35S phages were unlabeled. This means that DNA, and not proteins, were transferred from the phage to the bacteria after infection. The previous hypothesis that proteins composed hereditary genetic material was now weakened in favor of the DNA hypothesis. Ultimately, Hershey and Chase provided important evidence that DNA was the source of genetic information, and not proteins, as was widely believed at the time. Their findings would soon be verified by Watson and Crick in 1953.